Hello students, today we are going to discuss about semi-conservative replication in prokaryotes. In introduction, we will discuss the three proposed models of DNA replication. Replication is the process by which a cell copies its DNA prior to division. As for example, in humans, each parent cell must copy its entire 6 billion base pairs of DNA before undergoing mitosis. After the discovery of DNA structure by Watson and Crick, scientists started investigating on the overall nature of the process by which DNA replication occurs. Let us see how we define the models. As previously mentioned, Watson and Crick themselves had some specific ideas about DNA replication, and these ideas were based on structures of the DNA molecule. In particular, they hypothesized that replication occurs in a semi-conservative fashion. According to the semi-conservative replication model, the two original DNA strands, that is the complementary half of the double helix, separates during replication. Each strand then serves as a template for a new DNA strand, which means that each newly synthesized double helix is a combination of an old strand, which is the original, and a new strand, which is newly synthesized. Conceptually, semi-conservative replication made sense in light of the double helix structural model of DNA in particular. The complementary nature and the fact that adenine always pairs with thymine and cytosine always pairs with guanine. Looking at this model, it is easy to imagine that during replication, each strand serves as a template for the synthesis of a new strand with complementary bases being added to the order indicated. Semi-conservative replication was not the only model of DNA replication proposed during the mid-1950s. However, in fact, two other promising or prominent hypotheses were put across, which is the conservative replication and another is the dispersive mode of replication. According to the conservative replication model, the entire original DNA double helix serves as a template for a new double helix such that each round of cell division produces one daughter cell with a completely new DNA double helix and another daughter cell with a completely intact old original DNA double helix. On the other hand, in the dispersive replication model, the original DNA double helix breaks apart into fragments, and each fragment then serves as a template for a new DNA fragment. As a result, every cell division produces two cells with varying amount of old and new DNA. In order to define which type of model is followed by prokaryotic DNA replication, Messelson and Stahl did few experiments. In order to differentiate between the three models, Messelson and Stahl conducted some experiments. In their experiment, Messelson and Franklin Stahl were well acquainted with the three predictions, and they reasoned that if there were a way to distinguish old versus the new DNA, it should be possible to test each predictions. I mean about the three models which was proposed. Aware of previous studies that had relied on isotope levels as a way to differentiate between parental and the progeny molecules, the scientists decided to see whether the same technique could be used to differentiate between the parental and the progeny DNA. If it could, Messelson and Stahl were hopeful that they would be able to determine which prediction and replication model was correct. They thus began their experiment by choosing two isotopes of nitrogen, the common and lighter N14 and the rare and heavier N15, which we call as heavy nitrogen, as their labels, and technique known as cesium chloride equilibrium density gradient centrifugation as their sedimentation method. Messelson and Stahl opted for nitrogen because it is an essential chemical component of DNA. Therefore, every time a cell divides and the DNA replicates, it incorporates new nitrogen atoms into the DNA of either one or both of the two daughter cells, depending on which model was correct. If several different density species of DNA are present, they predicted each will form a band at the position where the density of the cesium chloride solution is equal to the buoyant density of that species. In this way, DNA labeled with heavy nitrogen, that is N15, may be resolved from unlabeled DNA, which is the N14. Michelson and Stahl then continued their experiment by growing a culture of E. coli bacteria in medium that had the heavier N15 isotope as a sole nitrogen source incorporated in ammonium chloride. In fact, they did this for 14 bacterial generations, which was long enough to create a population of bacterial cells that contained only the heavier isotope of N15 containing cells. Next, they changed the medium to one containing only N14 labeled ammonium salts as the sole nitrogen source. So, from that point onwards, every new strand of DNA would be built in with N14 rather than N15. Just prior to the addition of N14 and periodically thereafter, as the bacterial cells grew and replicated, Michelson and Stahl sampled DNA for use in equilibrium density gradient centrifugation to determine how much N15 from the original versus N14 from the new strand is being incorporated. For the centrifugation procedure, they mixed the DNA sample with a solution of cesium chloride and then centrifuged the sample for enough time to allow the heavier N15 and lighter N14 DNA to migrate to different positions in the centrifuge tube. By way of centrifugation, the scientists found that DNA composed entirely of N15 labeled DNA, which is the DNA collected just prior to changing the culture from one containing only N15 to one containing only N14, formed a single distinct band because both of these strands were made entirely of the heavy nitrogen medium. Following a single round of replication, the DNA again formed a single distinct band, but the band was located in a different position along with the centrifugation gradient. Specifically, it was found midway between where all the N15 and all the N14 DNA would have migrated. In other words, halfway between heavy and the light bands. Based on these findings, the scientists were immediately able to exclude the conservative model of replication as a possibility. After all, if DNA replicated conservatively, there should have been two distinct bands after a single round of replication. Half of the new DNA would have migrated to the same position as it did before the culture was transferred to the N14 medium, that is, to the heavy position, and the other half would have migrated to the new position, that is the light position, which is supposed to be the N14 bands. That left the scientists with only two options: either the DNA replication is semi-conservative, as predicted by Watson and Crick, or it may be dispersive. To differentiate between the two, Michelson and Stahl had to let the cells divide again, then sample the DNA after a second round of replication. After that second round of replication, the scientists found that the DNA separated in two distinct bands, one in a position where DNA containing only N14 would be expected to migrate, and the other in a position where hybrid DNA containing half N14 and half N15 would be expected to migrate. The scientists continued to observe the same two bands after several subsequent rounds of replication. These results were consistent with the semi-conservative model of replication and the reality that when DNA replicated, each new double helix was built with one old strand and one new strand. If the dispersive model were the correct model, the scientists would have continued to observe only a single band after every round of replication. After the Michelson and Stahl experiment, we conclude that the DNA replication undergoes in a semi-conservative fashion, and it was further studied in which manner it replicates. DNA replication can be studied in three stages, which we'll divide as initiation, elongation, and termination. In the initiation stage, the E. coli replication origin or EC consists of around 245 base pair and contains DNA sequence elements that are highly conserved among bacterial replication origins. 5 repeats of a 9 base pair sequence known as R sites and 3 additional DNA binding sites known as the I sites that serves as a binding site for the key initiator protein known as the DNA A, and a region rich in A and T base pairs called the DNA unwinding element. At least 10 different enzymes or proteins participate in the initiation phase of replication. They open the DNA helix at origin and establish a pre-priming complex for subsequent reactions. The crucial component in the initiation process is the DNA A protein. 8 DNA A protein molecules, all in the ATP bound state, assembles to form a helical complex in ORI-C. DNA A has a higher affinity for the R sites rather than the I sites, and binding R sites equally well in its ATP or ADP bound form. The I sites, which bind only the ATP bound form of DNA A, allows discrimination between the active and inactive form of DNA A. The tight right-handed wrapping of the DNA around this complex introduces an effective positive supercoil. The associated strand in the nearby DNA leads to denaturation in the AT-rich DUE region. The DNA C protein then loads the DNA B protein onto the separated DNA strand in the denatured region. Loading of the DNA B helicase is the key step in replication initiation. As a replicative helicase, DNA B migrates along the single strand DNA in the 5' to 3' direction, unwinding the DNA as it travels. The DNA B helicase loaded onto 2 DNA strands thus travel in opposite directions, creating 2 potential replication forks. All other proteins at the replication fork are linked directly or indirectly to DNA B. As replication begins and the DNA strands are separated at the fork, many molecules of single-stranded DNA binding proteins, also known as SSB, bind and stabilize the separated strands. And DNA gyrase, which is a DNA topoisomerase type 2, relieves the topological stress induced ahead of the fork by unwinding reaction. In the process of elongation, the elongation phase of replication includes 2 distinct but related operations: leading strand synthesis and the lagging strand synthesis. Leading strand synthesis, the more straightforward of the two, begins with the synthesis of primase, which is by DNA G protein of a short 10 to around 60 nucleotide RNA primer at the replication origin. DNA G interacts with DNA B helicase to carry out this reaction, and the primer is synthesized in the direction opposite to that in which the DNA B helicase is moving. In effect, the DNA B helicase moves along the strand that becomes the lagging strand in DNA synthesis. However, the first primer let down in the first DNA G and DNA B interaction serves to prime leading strand synthesis in the opposite direction. Deoxyribonucleotides are added to this primer by a DNA polymerase III complex linked to the DNA B helicase tethered to the opposite DNA strand. Leading strand synthesis then proceeds continuously, keeping pace with the unwinding of DNA at the replication fork. Lagging strand synthesis, as we have noted, is accomplished in short Okazaki fragments. First, an RNA primer is synthesized by primase, and as in leading strand synthesis, DNA polymerase III binds to the RNA primer and adds deoxyribonucleotides. Both strands are produced by a single asymmetric DNA polymerase III dimer. This is accomplished by looping the DNA of the lagging strand as shown in bringing together the 2 points of polymerization. The synthesis of Okazaki fragment on the lagging strand entails some elegant enzymatics choreography. DNA B helicase and DNA G primase constitute a functional unit within the replication complex, the primosome, which is known as the primosome. DNA polymerase III uses one set of the core subunit to synthesize the leading strand continuously, while the other set of core subunit cycles from one Okazaki fragment to the next on the looped lagging strand. The replisome promotes rapid DNA synthesis, adding approximately 1000 nucleotides per second to each strand, leading as well as lagging strand. Once an Okazaki fragment has been completed, each RNA primer is removed and replaced with DNA by DNA polymerase I, and the remaining nick is sealed by DNA ligase. DNA ligase catalyzes the formation of a phosphodiester bond between a 3' hydroxyl at the end of one DNA strand and the 5' phosphate at the end of another DNA strand. In the next process of DNA replication, we'll talk about termination. Eventually, the two replication fork of the circular E. coli chromosome meet at a terminus origin containing multiple copies of a 20 base pair sequence called TER. The TER sequence are arranged on the chromosome to create a trap that a replication fork can enter but cannot leave. The TER sequence functions as binding site for the protein TUS, also known as a terminus utilization substance. The TUS-TER complex can arrest a replication fork from one direction and prevent over-replication. Only one TUS-TER complex functions per replication cycle, so when either replication fork encounters a functional TUS-TER complex, it halts. The other fork halts when it meets the first. The final few hundred base pair of DNA between these large protein complexes are then replicated by an yet unknown mechanism, completing two topologically interlinked, which is known as the catenated circular chromosomes. DNA circles linked in this way are known as catenans. Separation of the catenated circles in E. coli is done by topoisomerase IV, which is a type II topoisomerase. The separated chromosomes then segregate into two daughter cells at cell division. In conclusion, we have seen that DNA replication is in a semi-conservative fashion as proven by Michelson and Stahl and as proposed by Watson and Crick, and we have seen that DNA replication is a process which requires a multiple enzyme interaction and can be divided into three stages, that is the initiation, elongation, and the termination. After the DNA synthesis, the cells will then divide to form independent daughter cells, which will grow and again carry out the process of replication before another cell division.